Hello, everyone. Let me start by thanking the Regenerative Viticulture Foundation for this challenge of sharing the stage for such an important topic. Thank you, Anne, Caine, Charles, and Emmanuel. Thank you also to everyone on the other side of the screen, wherever you are in the world. So in today's discussion, we'll have the immense privilege of listening firsthand to the insights and very practical results of a 3-year journey transitioning into regenerative organic. Caine Thompson from O'Neill's Vintners Distillers will be telling us all about it, and believe me, they will be sharing, and they've measured everything. But before we start our discussion for today, let me introduce Porto Protocol to those who are tuning in for the first time. We are the Wines Industry Climate Action Network. We believe in action rather than perfection, and our main goal is to foster peer-to-peer learning to accelerate the industry's response to climate change. We believe that by sharing experiences, solutions, best practices, lessons, and challenges, we can tackle this emergency much more promptly and efficiently. We are a click away, and we would love to meet you, so consider this an invitation to join us. So this climate talk, as I mentioned before, is led by the Regenerative Viticulture Foundation. We count with Anne Jones to host and moderate, Caine to tell us all about O'Neill's journey towards regenerative viticulture. And believe me, they have measured, as I mentioned, everything. Charlie Dubbe from Agrology, that was part of this journey, and Emmanuel Bourgignon from LAMPS 21, I'm sorry, Emmanuel, and La Repente to enrich this conversation. And without further ado, Anne, the stage is yours, and I'll disappear for now. Good luck, everyone, and thank you.
Many thanks, Marta. Thank you. Yeah, we're from the Regenerative Viticulture Foundation. Hopefully some of you will have heard of us. We are a charity that effectively is working with nature to inspire and support as many growers as possible on their regenerative journey through science, communication, and support for growers. So this is absolutely perfect for us in terms of working with the Porto Protocol. We love working with the Porto Protocol because we have all the same aims and we love collaborating. And so one of the great benefits of the RVF is our group of trustees, and Caine is one of those trustees. We're very fortunate to have him. He's very thoughtful, he's very measured, he's very scientific, but he's also very open and very collaborative. And that's why we're here today. So we'd love this to be quite discursive. We'd love to take all your questions. Please do put them in the Q&A as we go. If there's anything which you want to just contribute in a more kind of chat scenario, you could also use the chat. If you've got specific questions for Caine, Charlie, or Emmanuel, please put them in the Q&A and I will gather them together and either ask them at an appropriate moment or we'll cover them at the end of the session. And everyone's here to listen to Caine and not to me. So on that note, I will pass it over to Caine and thank you very much.
Thank you, Anne, and for that warm introduction and the platform, Marta and the Porto Protocol, and for the RVF as well. It's a pleasure to be here and looking forward to sharing the results of this 3-year trial and journey that we've been on. I'm going to share my screen here. All right. So just a little bit of history on how we got here. So O'Neill Vintners Distillers has been practicing sustainable wine growing as a pillar of O'Neill for 20 years. So to grow for O'Neill, you've got to be sustainable certified. We've worked with 200 plus growers across 15,000 acres, and to become sustainable certified here in California and the US. And then 4 years ago, we came across a certification called Regenerative Organic, and we heard about it through the Regenerative Organic Alliance and a pilot project that was being run by our friends at Tablas Creek 4 years ago. And the thought was, how about we start a project, a 3-year conversion to regenerative organic. I'd personally been farming this way for the last 20 years down in New Zealand under organic and biodynamic systems. And when we first came across regenerative organic, it was all of these great measures and tools and practices combined with a human cultural part as well to the system. I was like, wow, let's set up a trial. So I pitched the idea to Jeff O'Neill, our fearless leader, and said, hey, let's start a regenerative organic journey. And the pitch was, let's start with 1 acre a 1 acre control and a 1 acre regenerative organic plot. And Jeff being Jeff, he said, well, that's not much of a control and it's not much of a trial. How about we do a third of our state? So we set out a 50 acre conversion study, and I'm so excited and proud to present these results on behalf of O'Neill. The whole idea of the study was it was to be collaborative, it was to be open to industry. We hold these quarterly field days to really share the results and the learnings and this whole journey to regenerative organic. So this is basically the summary of the three-year trial to regenerative organic that we're looking forward to sharing with you. So we're going to go through the overarching purpose, location design, seasonal management programs, and then obviously into harvest results right through the 3 years, cost analysis as well. What does it cost to farm this way? In the winery, what is happening to the wines? This is really unique trial and that we've been actually making small microvin lots of each of the control and regenerative organic to see what is the actual effect on wine and wine quality in the long term as well. And then we partnered with Agrology to capture all of the data in regards to soil health, what is happening to carbon sequestration, soil moisture, organic matter, carbon and canopy. And we've got some great results to share with you on this front as well. And then next steps as a company in terms of where we're going with our regenerative journey. And at the end, we'll have plenty of time for Q&A, so please feel free to put all of those questions into the chat as we go through. So this overarching trial in 2020 was to really, the purpose was to establish this foundation of a true experiment where we're comparing conventional farming with regenerative organic, and what are the effects on soil health, yield, quality, cost, and finished wine over that time frame. So this has been a 4-year conversion from the initial idea back in 2020. It's an open industry trial, like I said, quarterly field days held throughout the year. And the idea of that was to bring everyone on the journey, because people are coming at regenerative farming at all different times of the year and also different times of their journey within their own production systems. So we found it being a really great way to onboard other growers and other industry members into regenerative farming. So the trial was set to ROC standards, so regenerative organic standards, developed by the Regenerative Organic Alliance. I'm sure many would have heard of the Regenerative Organic Alliance, as formed in 2017 by founding members Patagonia, the Riddell Institute, the largest organic researcher in the country, and Dr. Broner's all-in-one products. And they're really set to build a certification system around regenerative organic that encompassed everything about regenerative organic growing, and we'll talk about what those actually are. So the whole trial was set to these standards right from the outset. Organic CCOF is the baseline, which is a USDA standard for organic growing. So we started this whole transition. Our vineyards were certified sustainable. And so the idea was that we would certify and start growing organically and regeneratively organically right from day one at the conversion of the trial. So just to ground us in what is regenerative organic in regards to how we talk about it, it's this collective set of practices all around regenerating soil health and looking at the farm as a full ecosystem. This is at organic growing at the baseline, and it's adding and keeping vegetative cover. It's minimal soil disturbance in regards to minimal till, the addition of compost, and we'll talk about that shortly. Rotational grazing with sheep. I'm originally from New Zealand, so I can talk about sheep for a very long time. But our furry friends are pretty amazing in terms of what they do and offer. And then one of the most powerful pieces is the social fairness standard, which the ROA builds into their model, which is really unique and really powerful. So whereabouts are we? We're here, California, right in between San Francisco and LA, in Paso Robles, one of the great wine regions of the world. And Paso Robles is made up of 11 AVAs. We are here right in what we call the heart of Paso Robles, right there in the little red dot there. So we set up the trial, conventional control. This is a very standard conventional program. It's a sustainable certified system. Weed control, Roundup, Rely, Shark, generally are the herbicides that were used in conventional viticulture. Pest control, Wrangler, Movento, Intrepid. These are all insecticides that can be used for leafhoppers and mealybug and Torix moth. And then fungicides, a combination of sulfur, Champ, Rely, Quinstec, and Tibustar for powdery mildew control. Botrytis is not a major issue here, given how dry our climate is. And then a fairly standard nutrition program of zincs and calciums and phosphates. When we go into the regenerative organic program, organic is the baseline of a regenerative organic program in regards to the development. And so for weed control, we're using a Clemens undivined weeder I'll show you some images of this shortly, to keep a small area undivined, weed-free. And then pest control, we're doing two interesting ways of pest control. And we'll share this shortly. One is biocontrol with the use of mealybug destroyer. And two is mating disruption with pheromone traps. And so when you farm this way, you've got to think a little differently about how you control pest and disease, because you don't have synthetic options for insecticides. So we've had a really effective biocontrol program that I'm excited to share with you more. Then the fungicide program is sulfur for powdery mildew, and then oil for smothering for powdery mildew as well. Then nutrition, compost teas, preps, botanicals, combination of some BD preps, teas, and then some organic products as well. So trial design, so we're here in Paso Robles, the Geneseo AVA, 48 acres in total, 43 acres in year one, converted to regenerative organic, 5-acre conventional control. The trial set up on Cabernet Sauvignon, Clone 15, and then we had some Petit Syrah also converted to regenerative organic at the same time. So here is our beautiful estate here in Paso Robles. On the left-hand side is the trial site. So we've got in the area in the red, we've got the 5-acre control, and that area all to the right there is the block that was converted to regenerative organic in 2021. In regards to measurements, we take a 5-acre set and we do randomized replicated sampling from these different 5-acre lots. So it's a 5-acre control, 5-acre regenerative organic comparison, and then the rest of the block is obviously all regenerative organic as well. So in 2021, and we're going to go through all these results, but we started after 2021 to convert the rest of the estate here, because we're so encouraged by the results, into regenerative organic as well. So this estate here is 70 acres, and then we've got another 60 acres and another AVA called Estrella that in 2022, we started a regenerative organic conversion there as well. So in total, we've got 130 acres in different stages of transitioning to regenerative organic. So seasonal management, kind of what does it look like in the journey of starting to farm this way? In the winter, we bring in sheep for grazing. Sheep are amazing. We work with local farmers to rotational graze. So we bring in a certain amount of sheep per acre and then feed them across. And obviously, that's all about nutrient recycling, cleaning up the understory. And then after that, we put on a pretty heavy amount of compost, to help build back organic matter in the soil. So we're putting on about 6 tons to the acre, broadcast spread across the vineyard, so not banded, right across the soil profile. Then going into the spring, obviously we're putting cover crops down. There's a number of different preparations that we've been using as well, and compost teas. One great part of
I think we lost Caine for a second.
We just lost Caine, no problem at all. If we've lost Caine, Charlie, is there anything that you'd like to add on this point so far? And perhaps we could, I know there's been a question about downy mildew as well. So Charlie, I don't know if you've got anything that you can bring in to talk about the elements that Caine's mentioned so far.
Yeah, I mean, I don't think I have a ton to add at this point. I'm really excited to share some of the data that we have that sort of supported all this. Agrology was brought in. Yeah, I'll just kind of jump into an intro to Agrology while we get Caine back on. But Agrology was brought in year 2 of the trial. Agrology, we started really with climate monitoring, and then recently we've moved into soil health monitoring. So we monitor a variety of different factors that basically help quantify these changes in soil health. And looks like Caine is back, so I'll turn it back to Caine.
Yeah, sorry about that. We got kicked off there somehow.
No worries, Caine. Welcome back. As a link back in, actually, there was one question that was relevant to one of the slides you had just before, which was about the disease prevention. And we're just asking if you get any downy mildew issues.
We don't actually. Downy mildew is one of those diseases that we actually don't really get here, thankfully. So we're very fortunate in this dry, arid climate. That's one of the diseases that, like botrytis, we don't really battle that much. All right. Yeah, so seasonal management, one of the benefits of this program is their encouragement to work with the vineyard teams and in everything we do. And so there's an example here of some compost teas that we're preparing together and then applying them onto the vineyard. Cover cropping, really important part of a regenerative system. So we are planting a combination of annuals and perennials into the vineyard floor. And the whole idea is to add more diversity, to help cover the soil, protect the soil, and to encourage other beneficial insects into the vineyard as well. And the whole idea is to create these polycultural systems. So the biocontrol piece in regards to controlling pest, mealybug is the biggest pest we have here in wine grapes in California, primarily because it is the vector for a virus called leaf roll virus type 3. So the mealybug can feed on an infected vine and then pass that to an uninfected vine really efficiently. So without insecticides, what we're doing is working with a local insectary. And the insectary is the oldest insectary that's been around for 100 years in California. And they incubate the predators for us in terms of what we're looking to control. And so there's a predator called Cryptolaemus or mealybug destroyer that specifically targets mealybug. So they'll incubate the mealybug, and then we work with the company UAV IQ that specialize in the dispersion of the biocontrol agents and predators. So on the left here, this is the mealybug destroyer being put into the hopper here. And then these flight paths are set up and down the vineyard rows, and a certain amount of mealybug destroyer, a drop per acre, based on our pest and disease monitoring. And the beauty of this technology is that if we have a certain hotspot in an area of mealybug, we can put more mealybug destroyer to help control the mealybug in those areas. When we started the project, we were doing about 4 applications of mealybug destroyer. Now we're down to two drops a season as the population, the natural population, now starts to develop. So going into summer, so this is an example of the Clemens machine that we're using for undervine weed control. So we get really good control with the Clemens. And then again, through the summer period, a combination of the compost teas and preparations that we're using on the soil and then also on the canopy as well. So going into pre-harvest, so this is 2021, and this is pretty consistent in terms of what we're seeing throughout pretty much every season now, apart from one of the seasons that I'll show you. But if we've got conventional control here on the left, we've got the regenerative organic on the right, and we're seeing in general a bigger, robust, stronger canopy, a darker green leaf. We're seeing water hold in the soil more in the regenerative organic side, and we're seeing this bigger response. And for us here in Paso Robles, when it's so warm, this larger canopy is helping actually protect the fruit. It's providing some shade onto the fruit. It's lessening dehydration and shrivel. And it's providing like this really big engine for the fruit. Going into 2022, the same thing. Bigger, stronger, more robust canopy. You'll see the cover crop at this time of year in Paso Robles. I mean, we've had no rain from spring all the way through summer. The cover crops like self-terminated here by this point. Pre-harvest 2022, the same kind of visual here, larger canopy and a more robust compared to the conventional control on the left. Then in 2023, not as large of a vigor response, fairly similar in regards to look and feel of the canopy. And then this is right pre-harvest, is like the day before harvest. A little difference in regards to the color and condition of the leaf on the regenerative organic was more turgid compared to the control there. You can kind of see a darker leaf that's in better condition than the leaf on the left there on the conventional control. And then, this is just the day before harvest as well. And then this season, 2024, I just shot this a couple of days ago. We're back into a vigor response. We're probably a month out from harvest. Slightly larger canopy, bigger, denser, darker green leaf as well. So yield. So we harvest all of these lots separate. Everything's weighed separate. Everything is picked on the same day. All analysis is done at exact same time. So in 2021, with that larger canopy, we actually had a slight yield increase, a 14% yield increase. And that's not because there was more fruit on the vine. What it was that there was a canopy that was helping provide more shade, so there was less shrivel and dimpling of the grapes in the regenerative comparatively to the control. So we had more intact fruit, which was really interesting to see. In 2022, really dry year, yield effect more in line of about 3% increase, like small yield impact. And then in last year, 2023, a slight yield decrease in the regenerative side. So it's kind of bounced around a bit. And then, we look forward to seeing what 2024 is going to provide. And that's certainly something that going through this conversion, what we will be doing is continuing this program for the foreseeable future so we can track all of these measures for a longer period. So Brix at harvest, as you'd expect, kind of has followed the tonnage of the yield, so slightly less in the regenerative 2021 compared to the control, same in 2022 because you have a slightly larger crop, and we're also picking on the same day as well, so we bear that in mind as well. And then, as you'd expect in 2023, slightly higher Brix in the regenerative side, with there being a little bit less yield there as well. So when we take everything into the winery, we follow a native fermentation protocol, low amounts of KBS sulfur, obviously no oak, enzyme, tannin, or anything added. Pump overs once a day. No inoculation for malo, 100% French oak. And then there's a whole bunch of different parameters that we're measuring throughout the winemaking process that we'll talk about before we go into some of the soil data. So we're making wines every year from the blocks, doing a control, obviously at the regenerative organic from each year. So we're comparing and contrasting each year. So what we found really interesting around soil health and laddering into ferment health and quality of fruit within the fermenter. So 2021 yeast available nitrogen of the ferment, pretty much the same. And then we go into 2022, and we've had a 13% increase in YAN levels. And then that was followed in 2023 with a 17% increase. So what we're seeing here is healthier soils, healthier fruit, and yeast available nitrogens corresponding to that. Color is another measure that we look at in winemaking. And this is kind of bounced around a bit. General color measures here, obviously the control there in the yellow regenerative side, organic, and on the right. Slightly less color in 2021, and 2022 that was reversed. I mean, these are still big, dark, deep red, almost black Cabernets here in Paso Robles. And then 2023 kind of up and down a bit as well. Then we go into post-harvest. We're doing some barrel compound applications to the soil. And then we're bringing the sheep back in for this rotational grazing post-harvest as well. So cost, what does this all cost over time? And so the first year when we look back to 2021, we had new machinery coming in, operators were kind of learning to farm this way. We were doing more drops of the biocontrol agents. We were doing 4 back then. And so we were looking at about a 10% increase in the cost of farming back in 2021. But because we had that yield increase in 2021, it actually translated to a 3% decrease per ton of regenerative organic fruit transferred based on farming costs and yield to the winery. 2022, really low yields, a little bit more efficient in regards to our farming, so a 9% increase per acre in farming. Small yield increase in 2022, but resulting to a 6% increase per ton in regards to farming this way. And then 2023, certainly getting more efficient in our ability to farm under regenerative organic, a 7% increase in the cost per farm this way compared to the conventional control. And our yield was a little bit lower in 2023, which resulted in a net increase of 14% in regards to farming under a regenerative organic system. And with that, we're going to go into some of the soil analysis data that we have been capturing with Agrology. We've got some really great technology in regards to their Arbiter system that Charlie's going to walk us through. And we're capturing all this data from soil moisture to CO2 and canopy, carbon concentrations in soil, temperature differences between the control and the regenerative organic. And so we're excited to share some of these results with you. So I'll pass you on to Charlie here that can explain the sensors and what they're measuring.
Thanks so much, Caine. My name's Charlie Dubbe. I'm the head of regenerative partnerships at Agrology, and it's been such a pleasure working with Caine and being part of this trial. As you all can sense, doing them side by side is just such a phenomenal thing because in terms of gathering data, we have, this amazing, very clear side by side to look at. And so, I'm excited to present some of the data. I think a lot of it's quite compelling and makes a very strong case for the potential for regenerative viticulture practices. So Agrology, our system really is focused on measuring sort of the carbon cycle on farm. We definitely are focused on carbon sequestration, but we also know, and I think that probably most people in this webinar would agree, that carbon sequestration is actually just one of the positive ecological benefits that come from a regenerative system. And it's important not to get what they call carbon tunnel vision, right? There's these other ecological and operational benefits that come from regenerative agriculture that are perhaps just as important. In these systems, we are seeing improved soil structure because of the better aggregate. When you have more soil biology, more bacteria is creating the microaggregate, and then the fungi is binding those microaggregates into macroaggregate. That's helping with better pore space. Better pore space allows for better water infiltration. All the while, you're increasing your soil organic matter. So we're seeing improvements in the water holding capacity. We're also seeing better nutrient cycling. And so that allows for better nutrient availability. When we were looking at the photos of the vines and they were more green and larger canopies and more turgid, all of that has to do with that improved nutrient cycling, right? It's not like there's all of these additional nutrients that are given in terms of a synthetic form to that block. It's really allowing the soil's natural nutrient cycles to start to sort of heal and revitalize. And then another really important aspect of regenerative agriculture is when you create a more active soil microbiome, you're actually enabling the plant to sort of defend itself better from pests and diseases. We've seen there's lots and lots of research that shows how a stronger, healthier soil microbiome allows plants to have an increased production of these compounds that are called secondary plant metabolites. Secondary plant metabolites are they are phytonutrients. They are chemicals that the plants use to defend themselves from both biotic stresses and abiotic stresses. So things like, droughts or heat waves. What's really interesting is these are also the compounds that give wine its unique flavor and its unique profile. So these are compounds like tannins and all of the phenolic compounds, and the list goes on and on and on. All the flavonoids, all the terpenes, all of these things. And then they are all a way that the plant can defend itself. So not only do you have less pest pressure and less damage from pests, but you're also, in the end, creating a higher quality wine. And so that's something we're really interested in digging into a little bit more. And I think we're just starting to really scratch at that question. So yeah, again, just to kind of explain how the system works. So we measure CO2 at the surface of the soil, and then we also measure CO2 concentrations how the CO2 is being absorbed in the canopy. Overall, we really like to say that, we want to focus on this thing called the liquid carbon pathway. So the liquid carbon pathway is a term that Christine Jones came up with. And basically, there's two ways that carbon enters soil, right? There's sort of the decomposition route, which is this route on the left, a really great way to build soil health in the short term, but you're only getting about 5 to 10% of that carbon turning into humus, which is the long-term storage. And so this would be like mulches, adding compost, all the ways that you're actually physically adding carbon into the system. The more long-term way to build soil carbon and to build soil health is through plants, through photosynthesis. And that's that liquid carbon pathway, where carbon is being absorbed by the canopy and then brought down into the soil and released as a root exudate. And our system can actually pick up on these differences and pick up on these things. So, Caine, if you can go to the next slide. So this is kind of looking at one of our metrics that we use to assess overall soil health, which is called soil respiration. Most people are very familiar with probably the Haney soil test, right? The Haney test is where you take soil, you bring it to a lab, they dry it out, and then they remoisten it, and then they measure how much CO2 comes off of this soil sample over 24 hours. The total amount of CO2 that comes off of it is indicative of the amount of biology in that soil. Our system is really cool because it's like a continuous Haney test. So we're taking CO2 measurements right at the surface of the soil in a little soil chamber, which is like a little cup. And we're doing that every 10 minutes throughout the entire day. So what we're looking at here is sort of a continuous Haney soil test throughout the entire year. The green is the regenerative block and the blue is the control or the conventional block. As you can see, just way better activity, right? Obviously in spring, we see a lot more activity because things are moist and the temperatures are sort of more ideal. And then things start to level off as we get deeper into summer, although we did see a very nice spike at the end of July. But yeah, the pattern that we're seeing here, right, is just an immense amount of additional biological activity in the regenerative block. No big surprise. So how does this look in a day-to-day? What do these patterns look like kind of across a single day? It looks like this. In this graph, the green line and the purple line are regenerative blocks. You can see in general, they kind of follow the same pattern, but then we see this huge spike in the green line. What's the difference there? The green, the sensor that was the green line was placed right at the base of a vine. So we're actually seeing the influence of root exudates. And we've seen this in a lot of our different regenerative vineyards that we work in. In the morning, starting around 8 a.m. as photosynthesis kind of kicks on, we see this huge spike in biological activity. And this is a result of, as I said, photosynthesis kicks on, and then the plant sort of is signaling the microbiome, hey, I'm awake, it's time to start cycling nutrients, and releases this huge flush of exudates and respiration. And then we see that as the resultant sort of spike. We know that it has to do with the amount of sunlight, because on really cloudy days, you'll get these smaller spikes. And then the last thing we want to look at in this graph is the orange line that is down below, that is pretty much a flat line. That is the respiration that is coming out of the conventional block. So the other thing we do, and what's interesting about our system is oftentimes for people who are a little bit more unfamiliar with the regenerative sort of system and how carbon cycles in these systems, they say, okay, but more soil respiration, isn't that a bad thing? Doesn't that mean there's just more CO2 going out into the atmosphere? And although sort of on initial take, you would think that would be the case, we actually see in these systems that the regenerative canopy uses all that CO2. So it's channeling more CO2 into the microbiome and feeding it through root exudates. There's more biological activity, which results in more respiration or CO2 coming out of the soil pores. But then all of that is reabsorbed by the canopy. And that is clearly evidenced in this graph right here. The green line is the regenerative block, and the blue line is the conventional block. What we're looking at is overall CO2 concentration. So I don't know if you can see it, but on the left side, right around midway through the blue line is 420 parts per million. That is or I think we're around 420, 421 now. That is like the baseline atmosphere at any one time in any place. If you took the average of the whole globe, we're at 420 parts per million. So you can see the concentrations of CO2 in the regenerative block are significantly below that. And we actually saw 10.7% more CO2 being absorbed out of the atmosphere through the canopy in the regenerative block. Next slide, please. Why is this? Well, it's pretty obvious in this photo. If we can go back to the photo of there's clearly just more photosynthesis happening here, right? It's a larger canopy. There's just more solar panels absorbing more CO2. Now we can go to the water holding capacity. This is another really exciting one. So we know that for every 1% of soil organic matter that you add, you are increasing the soil's ability to retain 20,000 gallons per acre. And we really saw this. We saw this, very clearly throughout the 2023 season, and we're seeing it in the 2024 season as well. In this graph, we're looking at just soil moisture patterns. So they had the exact same irrigation. There was no difference there, but we consistently saw higher soil moisture levels in the regenerative block, which is the green line, versus the blue line, which is the conventional block. Over the entire season, if you average it, we saw a 13% improvement in water holding capacity in the 2023 season alone. And I think we're going to continue to see that grow. Next slide. So this is looking at canopy temperature. This is a really exciting one, and I think it's very interesting. In Paso Robles and in a lot of wine growing regions, there's this increased concern that it's actually going to be too hot to grow high quality wine, right? We're seeing desiccation, we're seeing dehydration, we're seeing all of those volatile compounds that we really need for high quality wine. They actually are volatilizing because it's so hot. we saw in the regenerative block, and keep in mind, the two sensors are only about maybe 200 feet away from each other. These are in the exact same climate. The only difference is the management practices. We saw throughout the entire season cooler temperatures in the regenerative canopy. And that's for a few different reasons. So number one, the soil is covered and it's armored. So we're not getting that refractive heat where the sun is bouncing off of bare soil and then jumping back up. It's covered in, even if it's not covered in a living cover crop, it's still armored by that desiccated and that dry cover crop, which is really reducing the amount of heat that's coming up and refracting off of the soil. The second reason is because there's more evapotranspiration, right? When plants photosynthesize, they release moisture into the air, and it's like nature's air conditioner. And we saw, as you can see here, there's pretty clear differences in temperatures. So this was during the harvest period in 2023. The green line is the regenerative block. Again, the blue line is the conventional block. And as you can see, huge differences. So, for example, on October 6th, we saw the regenerative block had the high of 98 degrees Fahrenheit, whereas the conventional block topped out at 106 degrees. And that's a really key difference, right? You all know about this much better than I do, but my understanding is right around 100, 102 degrees, that's a pretty critical threshold for vines. And you really don't want them going above that because of, again, all the desiccation and dehydration. Another really key stat on this one was that the regenerative block, the average canopy high during this month of October was 87 degrees with 1 day over 100. Whereas the conventional block, the average high was 94.3 degrees with 8 days over 100 degrees. Again, one cool thing about agrology is that we're actually measuring the temperature right in the canopy. So like in the same place where you're having your bunches of grapes. So it really allows you to see what's going on and what the conditions of the grapes are, which is what we really care about. Next slide. You can go to the next one even from here. Okay, so this is looking at soil temperature in the control versus the regenerative. One thing to note here that's really important is microbes don't like to be cooked. You know, at about 85 degrees, if you're having that top 6 or 8 inches of soil above 85 degrees, you might not be seeing dieback, but you're definitely seeing the microbes aren't proliferating as they could if the temperatures were cooler. And again, as you can see, not having that soil armored, having it be bare soil without living roots, much hotter conditions. So I don't think I have any key stats on this one, but you can see, especially on those hot days, you know, there's 5-degree difference, and we're seeing a lot of difficulty basically in the microbes. It's not the ideal conditions for them. And so that's not only creating, it's sort of like a vicious cycle, right? Where not only do you have less microbial life already because it's in a conventional system, but it's getting baked. And so you're seeing increased depression of those microbial communities. So what are the takeaways here? My big takeaway is that we can do landscape level cooling. It's possible with a shift in vineyard practices. So not only can we cool the nanoclimates around our vines and around our grapes, which obviously we care about from a business standpoint, but from a larger standpoint of sort of collective action, we can also cool our local watersheds and our bioregions. If you can get all of your neighbors together and everyone is, you know, doing the type of thing that Caine is doing, you can see how you could actually literally cool your bioregion through living plants and through boosting the microbial function and the diversity in our soils. So this is great because this is not only local climate repair, which is really how we start to repair the global climate, you know, repairing the carbon cycle, repairing the hydrological cycle, all within our bioregions, but it's also allowing us to continue to grow high quality wines. Overall, we saw a 7.8% decrease in the average daily temperature during that critical harvest period in the regenerative block. So it's giving real, I mean, this is talk about ROI. This is like as clear as it gets. Next slide, please. So this is just looking at some key temperature differences. You know, we already talked about some of these. Again, the average canopy high in the regenerative block was 87, whereas in the control block, it was 94. We saw 8 days over 100 degrees in the control block or the conventional block versus just 1 day in the regenerative block. The average volumetric water content in just this month of October was 19.1% versus 17%. Again, remember, this is at the very end of the season, so the soils are very, very dry, and even a 2.1% difference is really huge. And then overall, as I mentioned, a 7.8% decrease in temperatures in terms of the average daily high in the regenerative block. Next slide. So how does this all tie into carbon sequestration? We know that more living plants and more photosynthesis means more carbon sequestration, especially when paired with more biological activity. Those are the two things you really need for carbon sequestration, right? We can get lost in kind of all the complexities of the labile fraction versus the recalcitrant, but really when it comes down to it, living plants photosynthesizing plus a very healthy and vital microbiome, those are the precursors for carbon sequestration. Carbon enters the soil in two ways, as we talked about. There's the liquid carbon pathway, where much more of it is actually sequestered for the long term, versus if you're physically just putting carbon into the soil, it's great, but the microbiome really churns through it very quickly. It's like feeding a furnace. You need to just keep feeding it. And we saw clearly that we have these preconditions for carbon sequestration. We saw an increased amount, a significant amount of carbon being absorbed by the canopy of the regenerative block, which means more carbon is entering into the soil system via this liquid carbon pathway. And that's on top of the 6 tons of compost that Caine is applying. For the month of October, you can see here in the graph, we're looking at this is soil microbial biomass carbon. So this is the carbon that is actually all of the, the accumulation of all of the bodies of the microbes. And then kind of jumping back into how much CO2 is being absorbed by the canopy. So this was for, again, the harvest month last year, October 2023. We saw the accumulated CO2 that was absorbed by the canopy. We saw 26 parts per million in the control block versus almost 1,400 parts per million in the regenerative block. So the regenerative block is absorbing basically 200% more CO2 than in the control block. Next slide, please. So this is looking at kind of directional stuff, changes over time, which is really, we know we're going in the right direction, but like, how fast are we going in the right direction? So this is from August 2023 to this month, August 2024. We saw a 9.1% increase in microbial biomass carbon, which is just one fraction of carbon, but I always view carbon as kind of a cascading thing. I like to use the metaphor of like money goes into your checking account and then your savings account and then your retirement account. And the microbial biomass carbon is sort of the checking or the savings. It's the first phases. So we saw a 9.1% increase in microbial biomass carbon over that year, whereas we saw a 2.8% decrease in microbial biomass carbon in the conventional block. And then you can go to the next slide and I'll talk a little bit more about the totals here. So as part of Agrology service, we like to look at soil health through a bunch of different metrics. So we also use soil tests. And we did a soil test just a few months ago. This revealed that the regenerative block had 2.4% total organic matter versus 1.4% in the conventional. The organic carbon, and this was done in the Haney method. So this is, I believe, water-soluble organic carbon is 1.5% versus only 0.86% in the conventional. You can see also more organic nitrogen, and then you can kind of see the carbon and nitrogen balance there, which is a really important ratio to keep your eyes on. The Haney soil respiration. So this is the same kind of data that Agrology does every single day, but this was in the laboratory. We saw 30.6 parts per million in the regenerative block versus 17.5 parts per million in the control block. Again, I just talked about, we saw that improvement in soil microbial biomass, 9% increase versus a 2.8% decrease over the year. And then the Haney health score. So this is a health score they give kind of looking at all your different metrics. We saw 7.8, which is a really good number to see, versus 4.7 in the control block. And then a really important one that everyone really cares about, we don't want to discount the importance of carbon sequestration. With these soil tests, if you run the numbers, you see a 12.23 ton difference between the regenerative block and the conventional block. So after 3 years, again, we'll go back and do more soil samples to reconfirm this number, but it looks like Caine has accumulated an additional 12 tons per acre using these regenerative viticulture practices. And I believe that's it for me. Let's see if there's one more slide. Nope, back to you, Caine.
Awesome, thank you, Charlie.
That was amazing. I might jump in with a few questions here because we've got some great ones. And what I might just do is ask a couple of the ones to Charlie that are most relevant to what you've presented. Then I'm going to get an opinion from Emmanuel, see if he's got anything to add. And then we're going to go back to Caine, both for his wrapping up and the next steps. But I just want to drop in a couple of questions. One of the questions was about actually this carbon sequestration. And Caine, are you actually using your carbon sequestration for any commercial value at the moment?
Not at the moment. We're not. We're looking into that, but at the moment we're not using those we're not selling them as credits or anything like that. But what we are looking into is, can we use that to offset our wider carbon footprint as a organization?
Amazing, thank you. I think the word is mitigate, not offset. Just for clarity.
Yes.
And then there was a quick question for Charlie on actually which instrument are you using to measure the soil activity? The one where you had those spikes that came in every morning with the photosynthesis. What instrument are you using to measure that?
Yeah, so it's a thing that Agrology has invented. So soil chambers have been used by academics for a really long time to study exactly this, but they're typically made by companies like Lycor, and they cost like, I don't know, 30 to 40 grand, and you need a PhD student to run them. So Agrology, our big sort of contribution to the space is we took that scientific instrumentation and we've just commercialized it. So we've made it a lot cheaper and a lot simpler. It's called Meter Group, and they have kind of a CO2 sensor that we put on top of a soil chamber. The soil chamber looks like that. And essentially what it is the soil chamber just prevents mixing with the wind. And then it has little vents, and we know how quickly air vents out. And so based on that, we can basically you look at for CO2 concentration. So you just look at how many parts per million are there. And then you convert that into what's called carbon flux, which is by mass, how much CO2 is going in and out of the system. I'd also be happy to chat with anyone who has more curiosity about our technology or sort of the sensors we're using. Feel free to email me. My email's charlie@agrology.ag. And we love chatting with folks about what we're doing.
Thanks very much. Sorry to interrupt you. Back to you, Caine. You can finish this bit off.
Thank you. So really encouraging results all around. There's definitely a desire to continue the trial. So it was originally set up as a 3-year conversion trial, but at O'Neill, we want to continue this for the long term because as we look to the future, we do think regenerative farming can and will change the world, but with more growers onboarding into this form of farming, more companies looking at this, we want this trial to be an area that people can come learn, see the results, and then hopefully take that step within their own properties or organizations to make that change. So we'll be continuing this trial for the long term. Like I said earlier, it's expanded into 130 acres of estate vineyards, and then all company vineyards, 800 acres plus, now have conversion plans into regenerative as well. We got our organic certification in January 2024, and we just completed our ROC audit in August 1st. So we're pending ROC certification. Should get notification of that really, really soon. Yeah. So with that, open the floor to wider discussion. And
absolutely, we've got some really great questions. There are a lot about the viticultural side, but I'm going to come back to them in a minute, because first of all, I'd like to hear from Emmanuel, and I'd like to hear what his perspective is and any specific questions he's got from his technological viewpoint on the other side of the planet.
Sure. Thanks, Anne. Great stuff. Great work. Very, very interesting to follow this presentation. I had myself also a few questions just to make sure I got things right. Are both blocks receiving 6 tons per acre of compost? or is it only the regenerative?
They both are. They both
Sorry?
are. They both are. They're both
Yeah,
receiving
they
compost.
both are
Yeah.
receiving. Yeah. And
Yeah.
the other thing for the compost, what was the citrate ratio you're getting on that compost?
The carbon nitrogen ratio of the compost.
Yeah. Roughly.
I'm not sure what it was.
Okay.
I'd need to look back at the test. Yeah.
Yeah.
It's the same across both blocks.
Okay. Yeah, great. So clearly that's what's powerful, is that you see how just adding the carbon, and Charlie pointed it out, but just adding the carbon to a system is not the solution really. It has to be a little bit more complex than that. I noted the stronger canopy, which is a big tell sign to me. It wasn't mentioned, but bigger canopy means also more shade to the soil, and in systems like yours, regarding microbial activity and also carbon flux, the having shaded soils increases greatly the activity of the microbial community. Like in places like Africa, or like places big droughts, we work underneath the trees to get better crop production and so on. So I get that from those bigger canopy, and overall, to me, it's a very elegant way to look at the carbon flux, measuring what's going on in the canopy and what's coming out of the soil. It's great. Where did I write down a few things? Yeah, the thing I think will be really good in the coming years is, as you gather data, and I get that from the past and from collaborating with many wineries for a long time. I'm pretty sure you're going to see like a stronger buffering effect of your regenerative viticulture regarding the climate. Like, you showed us data for 21, 22, and 23 with some climate clearly fluctuation regarding the yields and so on. I think as you run the trial, you would see, if you keep the control anyway, you would see that having those richer, more biodiverse system, you clearly have a buffering effect on the climate extremes, I would say. So that was this, and yeah, an overall big hands up and clap for all the work. Very exciting stuff. Yes, just one thing. Did you guys maybe looked into if there is any difference in the bacteria to fungi ratio between the regenerative and the control block, or not at this stage?
Not at this stage, but it's something that we definitely want to do. Part of the ROA framework is looking at like diversity within the vineyard. So we've done general diversity tests in regards to just living life.
I wouldn't be surprised if you find a stronger, larger fungi community within the regenerative than in the conventional, because that's one of the downside of this conventional management, is that we tend to encourage mostly the bacterial community, and the fungal community tends to decrease, or sometimes almost disappear. And I think the fungi are much stronger. I'm talking very generally, but if you go into the basidiomycetes and the ascomycetes and so on, they are much stronger at sequestering the carbon into the soil, but also at increasing like you talk about the liquid pathway of the carbon. When you have the fungi in the system, you speed up the speed up of carbon. And the other thing with those fungal community regarding the water holding capacity of the soil, sure, the carbon is having an impact, a strong impact on, but the microbial communities also hold water within their cells, within their structure, and that impacts the water holding capacity of the soil. But the ones that are the best at it by a long shot are the fungi, the Basidiomyces, because they have like certain type of cells and what we call aquaporins, certain type of proteins that are like super water retention system. And when you have these guys in the system, then you find, especially here in California, where you have dry soils, the differences are much stronger. They really stand out really quickly within a few years.
Yeah,
totally.
I definitely want to do a PLFA and maybe a full like metagenomic analysis next year on these sites, because I think, like you said, Emmanuel, I imagine we're starting to see some pretty distinct differences in that ratio. And also, as you said, like the mycorrhizal fungi are so important for not only that liquid carbon pathway, but also for transporting that water to where it's needed, right?
Yeah.
And transporting the nutrients to where it's needed. It's absolutely critical.
I'm going to have to jump in here, not because I want to, because this is fascinating. We have gone over the hour slightly, but given we've still got quite a lot of people here and given this is being recorded, I'm going to try and get through a few of the questions. If people want to drop off, you're very welcome to come back and watch this at a later date. But if we can get through some of these questions now quickly, I think it'll be really beneficial just because of the level of expertise we have on the call. So related to that last conversation, we've had a couple of questions about soil compaction linked to water retention, and also whether there's drip irrigation and what role that plays for you, Caine. So I'm going to try and loop these questions together so we can answer them quickly. But how do you address soil compaction, and how are you measuring soil compaction, and does it relate to your water holding capacity?
Yeah, completely. So we're trying to limit the amount of tractor passes. Obviously, we think the utilization of drones for biocontrol is a good example of minimizing compaction and restricting the amount of tractor passes that we're doing. We certainly are seeing with cover cropping, composting, the soils, they're definitely getting spongier. Like when we started, they're getting softer. When we started the project back in 2020, you'd take a spade and you couldn't even get the spade in the soil. It would like bounce off. Now it's open, it's more friable, there's density, there's complexity within the soil. It is dry here. We're obviously we get our rainfall in the winter, so we're trying to stay off land with equipment at that time. So those are some of the techniques we're trying to use to minimize compaction. But that would be something interesting to look at, like measuring the differences in compaction between the control and regenerative. We haven't measured that yet. But the beauty of having this trial in place is that any new thought that comes in terms of things that we could be monitoring and measuring, we can take that, and because we've got the control of how it was basically 4 years ago, we can compare with our regenerative organic practices and start getting some information around these things.
On compaction, let's not forget when you have a system where, because you have more microbial bioactivity and that basically you build up the pore space within the soil, a soil that has a lot of pore space is a little bit more resilient to compaction than one that doesn't, especially when the soil is at field capacity. That is usually where we do the most damage, because if you have a soil that is already compacted with a little pore space and you are at field capacity and you drive on this soil, you transmit the compaction really deep into the soil. While if you have a soil with a lot of pore space, it can buffer this compaction. I mean, I'm not saying you can drive on it as much as you want, but you have a little bit more room, and the system can accommodate it better and can repair itself faster. Hence the word resilience, which in this case is really the right word to use.
Yeah,
brilliant. Thank you. And then I've got a question about, given your lack of disease pressure there in your climate, what's the reason for your undervine cultivation?
It's weed control. It's solely for weed control. So undervine cultivation, we only need to do about 2 passes per year. in the early spring. So we sheep graze for as long as we can. We've got a fairly high wire, so we can sheep graze.
When do you bring the sheep in? Sorry, that was another question.
After harvest, and then we block feed them through post harvest, and then we get them out before the winter rains ideally start, and then bring them in again early spring, and get them out mid spring.
Amazing, thank you. And how do you handle your cover crop residue, if you have any? I didn't see very much, but so what sort of tillage practices are you using, if any?
We don't. So the cover crop we just leave as permanent cover, and so the sheep will graze it down, but then we just leave it and just let it grow. And
then for your cover crops, so are you sowing every spring? What are you using? Are you trying to use native species?
Yeah, it's a really interesting topic. The majority of cover crops that are available, like commercially with the organic seed, is mainly annuals. So it's mainly annuals with a couple of perennials, but we're in the process of developing a native perennial diverse mix of 20 different species specifically for Paso Robles and California that actually can survive these dry, arid conditions. So we think there's a big opportunity there in regards to the development of that, because as you've seen in some of those photos, a lot of those annuals come at the peak of summer are kind of dying off, and you've got a layer of vegetation there, which is helpful for just mitigating some of the effect of the sun. But if we can have a living species that's alive right through, we think that's going to be a lot more beneficial. So more on that to come.
Brilliant, thank you. So we've got a couple more sheep questions, and I know people have so many sheep questions, so I'm not going to focus on sheep for too long, but just how high is your lowest wire?
The lowest wire, it's about lowest wire for cordon wire is about 5 feet on this block, so it's pretty high.
Perfect, thank you. And could you just clarify exactly when the sheep do come out of the vineyard? Is it after bud burst?
It's a little bit after bud burst, so we're about shoots 1 or 2 inches, because we've got that high cordon wire. We can actually graze for a longer period than what most can. But if you don't have that high cordon wire, then you have to get them out pre-spring, because they will chew the shoots.
Cool. And then I think in terms of the questions, there's just 2 more little topics to cover. One is where you have got additional costs for regen versus your conventional control. Where are those costs? What exactly are they? What are the things that are making it more expensive?
Yeah, so the 2 things that are making it more expensive, one is the drone drops. The biocontrol is definitely more expensive than insecticides. And then Undivine till is more expensive. It's just slower using the Clemens comparatively to herbicide. So those are the 2 areas. All other costs are relatively the same.
Amazing, thank you. And then just coming onto the final wines, there was a question about what's used to measure the colour, but I think some of the more technical questions I'm going to ask people to direct directly to Charlie. So his email address is in the chat. So Edwin, I think your question probably be best directed straight to Charlie, or else we are going to totally run out of time. But just a couple of questions that have happened around what the results are in the final wine, and if you've seen any quality differentials in blind tastings, or how you've measured that.
Yeah, so we do double-blind panels at the tail end of harvest with local winemakers. So winemakers come from all across California, and we do it double-blind, and the unanimous conversation and outcome is that the regenerative style, it's fresher, it's more vibrant, the more softer tannins, more elegant as well. There's not any form of cooked fruit that can occur if you get overexposed fruit, the kind of pruny, kind of jammy character that you can get if fruit gets overripe and starts to dehydrate. So it's been pretty unanimous that there's a quality improvement in regards to style across the 3 years. Yeah,
fantastic. Thank you so much. I mean, I think you've been brilliant. Emmanuel, over to Emmanuel. You've got another point.
No, I just had a question for Caine, because I forgot to ask it earlier on, was the soil, what is the soil texture? It's just...
So it's a calcareous base, and then it's a sandy loam texture. Yeah.
Do you have an idea what's your percentage of clay in your soil?
It's pretty low, the clay percentage. So it's mainly sandy loam.
Okay, okay.
Over a calcareous base, but small amount of clay, but certainly not a heavy clay.
That will be, we can talk about that in the future, but that will be in terms of the organic content you will be able to achieve in the soil. You will reach a maximum quite quickly. You won't be able to go really, really high in the organic matter because of this texture.
Yes. Yeah.
Amazing. Thank you so much. The remaining questions were about certification, and I think we could do another whole webinar on that. We do have a page on the RVF website if people are interested. But other than that, maybe Marta, that's something that we can pick up in due course and talk about certification separately. But I just want to thank Caine so much, and Emmanuel and Charlie. It's been incredibly informative and the most incredible set of data that I think most people will have had access to. I'm going to hand back to Marta now to close the session, but thank you all. What a great presentation and fantastic set of data. So thank you.
It was indeed. It is so important for other farmers, other vintners thinking of going into regenerative to really have data that they can count on to compare. So thank you very much for that. Thank you, Caine. Thank you, Charlie. Thank you, Emmanuel, on Porto Protocol side to come in and bring his view as well. Just before going, we will see you soon in London. We're at the Sustainability in Drinks on October 14th and 15th at an event that is co-organized by Anne Jones, by the way. Charlie, I know you wanted to announce an event as well, right? Just if you want just a second.
Yeah, thank you. Yeah, so we're doing another regenerative organic vineyard sort of webinar with the ROA and ROC organization directly. It's next Thursday, I believe, and I will find the link really quick and drop it in the chat. Let's see if I can do it before we end.
And remember, everyone on the other side of the screen, we are the Porto Protocol. This is an invitation for you to join us. Regarding this topic, we absolutely love working with the Regenerative Viticulture Foundation. That's why we're here today. But let me tell you that we also have an amazing project that Emmanuel is part of the leading team called Living Vineyards, in which we look not only at soils, but everything that is above and around ground, with more than 40 companies from 14 countries. So do join us, do join the Regenerative Viticulture Foundation, and remember that collaboration is key for us to tackle this big issue that is climate change. So see you maybe in London. Do write to us if you want to meet us. Do check out the Sustainability in Drinks website as well, and see you soon. Bye-bye. Thank you all.